you, John. So it's my great pleasure to introduce Mark Stitt, and we're running a little bit late. So Mark, if you would like to take off.
Okay. Can everybody see my screen?
Yes, looks good.
Okay, so this is a very great honor. It's also probably the most frightening talk I've ever given, because usually you can see the people, and now I can't. So what I want to talk about is how we've been using targeted metabolite profiling as a top-down approach to uncover interspecies diversity in the Calvin Benson cycle, basically how it's been adapted. And a little bit of introduction. We all know the Calvin Benson cycle is about 2 billion years old or more. And as far as we know, the reaction sequence hasn't changed since then. But there's ample evidence for variation in how the cycle operates. It's how it's regulated, how it's poised between cyanos and eukaryotes, between photosynthetic organisms that do and don't have a CCM. I mean, rubisco specificity is changing. And even between C3 higher plants, which differ in photosynthetic light responses and so on. And I've just listed some classic reviews on this topic. Now, most mechanistic studies of CBC diversity are focused on a single trait. They've gone in and, for example, they've compared rubisco kinetics or rubisco regulation across different species. There's also been some very nice work on leaf anatomy and ultrastructure, which I won't be touching on today, although it's behind some of our results. What I want to do is to introduce what I call a systems or top-down approach to look for interspecies variation without initially assuming what might be responsible for it. Now, metabolites have been measured for decades to study how metabolism is regulated. And so here's a sort of a sketch. We've got a lot of cars going through, and then we've got a lot of cars piled up. And of course, you could well deduce that here a traffic light has gone on. It could even well be a cow on the road. It could be the car has had a breakdown. It could be somebody is dancing in the middle of the road, but something's happening to hold the traffic up. And this principle is used in looking at metabolism. You perturb the system to decrease flux, and the enzyme at which flux is inhibited can be identified because its substrates rise, and often, I'll put it in parentheses, its products fall. And of course, the reverse is also true. If something happens to increase flux, then the substrates fall and the products rise. And we've asked, can metabolite profiling be used as a top-down approach to search for interspecies variation in how a pathway like the CBC operates? Basically, the difference, the balance, changes in the balance between different enzymic steps. Why bother? Well, I think it might get insights into the adaptation and evolution of the Calvin Benson cycle, and it might also help to think about what are the best strategies to improve photosynthesis in a given crop if we know where that crop is different to others. Now, all we need to do is to measure reliably and quantifiably all or almost all of the metabolites in the relevant pathway. Many studies have studied metabolites in the 1970s and 1980s, but usually only a few metabolites for technical reasons, and usually only one species per study. So we needed a method where we could measure pretty well all the metabolites. And for this, we use LC-MS triple quad, where we basically separate metabolites by chromatography on charge and size. Then in the first quadrupole by choosing the mass of that compound. Then we fragment it, and then you look for the fragments. And this gives us 3 filters to obtain a very specific identification of metabolites. So we get very nice peaks, clear and clean, but there are pitfalls awaiting. And this is the North Face, this is the Everest. As you're coming up to the top of it. There's about 2,000 meters drop there. What are they? Well, here's 3. Changes during harvest and quenching, changes during storage and extraction, and errors in analysis. And I'll just touch on these. Changes during harvest and quenching. The key point here is the pools in the Calvin Benson cycle are small, the flux is high, so the metabolites are turned over very rapidly. And most of the Calvin Benson cycle metabolites have a turnover time of less than 1 second. That means that if you put the plant into the dark for 1 second, you measure what the metabolites are after a plant has been put in the dark for 1 second. And so this means that we change the metabolites if you don't quench very quickly and without changing the conditions. And this slide just shows some of the approaches we've taken. Arabidopsis in a plastic box where you just tip a liter of liquid nitrogen on top of it. Or this is maybe the best one, where you actually have it within almost a gas exchange system, and you just drop a very cold metal column on it and freeze it. Or if you don't want to do that, two big, very large metal blocks, which you just use to crunch the plant, or a system which Marta Ludwig has built up, which we're finding very useful now. A point. These methods are not necessarily approved by the JXB. This is in the hands of the reviewers if they want to take them or not. No guarantee. Now, other problems are during extraction and analysis. Many metabolites are unstable. For example, a very simple example, sucrose is acid labile. So if you have a period of time in a weak acid, it's going to turn into glucose and fructose. Some enzymes are very tough and survive treatments, and the metabolites, false identification, overlapping signals, unspecificity, matrix effects, ion suppression, and so on and so on. These may well differ depending on the tissue, but also on the species. There's one basic thing to do. Lenin knew this. Faith is good, but control is better. So you extract your plant material and you measure the metabolite. You also get it from Boehringer or Sigma, no advertisement, and you measure it. You then tip the same amount of pure metabolite in as you're making your extract and assay. If A and B don't add up, if you get less than you expect, you have a problem. So to lose a metabolite can be unfortunate. Not to know you're losing it is carelessness. Oscar Wilde. And so Steffi Arevo synthesized a whole load of stable isotope labeled internal standards to allow us to really quantify and check our methods. So now we come on to the biology. By about 2018, in a whole series of projects and collaborations, we profiled Calvin Benson cycle intermediates in a whole series of species. And the idea came to throw this together. Now, this started with a very stupid-minded idea. We've got C4 species, we've got C3 species, and so we thought that these would separate, given that the Calvin Benson cycle in the C4 species has been operating in high CO2, this has allowed changes in Rubisco and so on. So we thought, oh, just get a nice separation between C4 and C3 species. In fact, we could just see by looking at the measurements that we had got some expected changes. Ribulose bisphosphate was lower in the C4 species because they have more efficient Rubisco, but a lower abundance of the Rubisco. This was expected. So we did the PCA, the principal components analysis, and the four species, which are green, and the C3 did separate very nicely from one another. But the C3 species were all over the place. Thought initially this is a pain, or actually this is interesting. This is telling us that the C3 species vary in how they're running the Calvin Benson cycle. Now this could be an artifact. For example, they were all grown in slightly different conditions to adjust to the plant. We might have different leaf composition. So let's look at this. There'll be more data coming later, but this just shows the metabolite profiles in maize and Arabidopsis at two different light intensities for maize, 3 different ones for Arabidopsis. I think you can see by eye that maize is consistent across light intensities, Arabidopsis is consistent across light intensities, and they differ from each other. And when we actually look, this plot actually includes maize at 2 light intensities and Arabidopsis at 3 light intensities, and you can see that these changes in the conditions are not altering the observation that the species group differently in the PCA. The second issue we thought about is different leaf composition. For example, higher protein, higher chlorophyll, and so on. This might lead to a shift of all metabolites, to systematic shift. So the approach that we took to look at this is basically normalize the data, not just on fresh weight, but also on protein and on chlorophyll. And then you look in, and basically we retain the separation of the species when we normalize them in different ways. The normalization we like the most is what I call dimensionless. This is, we just take the carbon we have in a given metabolite and we express it as a fraction of all of the carbon in all of the metabolites we're looking at. And this is this one down here. And you can see we have, again, a very nice separation. That this is basically telling us how is the carbon allocated between the different metabolites in the Calvin Benson cycle. So what reactions differ between the species? Small primer to thermodynamics in pathways. This is sort of a very bad analogy, Some enzymes catalyze irreversible reactions where the water is going from one level to another, and others, we have a little pool where the water is sort of swimming around and is mixing, and these are reversible reactions. And typically, the irreversible reactions are the ones that are usually regulated. Not always, but typically, they are the ones that are regulated. So now let's go to our data set. Now what we've done here is we've just asked for each pair of metabolites, are they correlated across the species? Are they unrelated? Or are they actually showing a negative correlation between the species? And this is shown in this heat map up here. Everything that is red is positively correlated. Everything that is blue is negatively correlated. And the white stuff is in the middle. And what we can see is, first of all, we have a pattern. We actually have a similar grouping in C3 and C4 species. It's a pain because the clustering also flips things the other way. But basically, we've got ribulose bisphosphate, FBP, SBP, and triose phosphates tending to correlate together, and the others tending to be in another group. And this is something which I'll go on to the next slide. When we look at which of these reactions, which of these reaction pairs, metabolite pairs are involved in a particular kind of reaction, most of the things which are correlated are reversible. Most of the things which are anti-correlated, negative, are irreversible. Or putting this in the context of the pathway, we usually get positive correlations between metabolites, which are involved in reversible reactions, and we have negative correlation or no correlation between enzymes which are catalyzing an irreversible reaction. So summarizing that, and this made me very happy, plants can't change the laws of thermodynamics, but they can change the rate, the balance between the irreversible reactions, as they can change expression levels or the regulatory properties or the characteristics of the proteins. So now I come on to another experiment. In this experiment, we took two plants, Arabidopsis and rice, and we harvested across their light saturation curve. And the aim here was to test more rigorously that differing metabolite profiles in C3 species are not just due to environmental conditions. And the other was to obtain a denser data set to learn more about what is diverse and also to see if anything is conserved. So this is the PCA, and I think you can see immediately that they've got different trajectories. That as you go a low light and darkness, they're the same, and the further we push up the light, the more different they become. Now, what's going on here? Shared is a general rise in many metabolites. This is captured in PC1. You can see that the weightings of all the metabolites are lying over here. So metabolites rise as photosynthesis rise, but they differ because in PC2, they're moving into different directions. And this, again, is the metabolites which are involved are down here. So what we next did is we got rid of the general rise of metabolites, because what we then went to is the dimensionless normalization, where we just asked what percentage of the carbon in the metabolites is where. So we got rid of the general rise, and then we asked for the relations. And what we see, they're just in totally different metabolic spaces. The two species, they're separated in PC1. Then we also see the lowest light intensity. This is this guy here and here. This is an outlier in both species, and this is a response which is caught in PC2, and I'll come back to this. It's driven by high SBP and high FBP. So now let's look into individual metabolites and metabolite ratios. So this is the raw data, and I think by eye you can see there are many differing and some shared responses. And I'll just pull out a couple of these. So these are differing responses. We have a lower PGA to DAP ratio in Arabidopsis than rice. Now this is where the ATP and the NADPH from the light reactions powers into the CBC. So what this is telling us is a shifted balance between the light reactions and the CBC to favor the light reactions in Arabidopsis. A second difference is we see the FBP to SBP ratio changes between Arabidopsis and rice. And again, this is consistent across the entire light range. So the balance between FBPAs and SBPAs isn't looking good for SBPAs in rice. And a third difference I've pulled out, pentose phosphate and ribulose bisphosphate rise progressively with light intensity in Arabidopsis, but they plateau at a fairly low light intensity in rice. So what this is telling it is, as light increases to drive CO2 fixation Arabidopsis ramps up, increases ribulose bisphosphate generation, whereas rice is ramping up ribulose bisphosphate utilization. What's conserved? I've pulled out two things here. One is that in both species, fructose bisphosphate to fructose 6-phosphate and sedioheptulose bisphosphate to sedioheptulose 7-phosphate ratios peak in low light and then decline. What we think is behind this, and I'll come on to, is low activity, probably post-translational redox activation or inactivation of these two enzymes in low light. Another general feature that we see is as you go up with the light intensity, the levels of triose phosphates, DAP, and other Calvin Benson cycle, which I haven't shown, they tend to go up very sharply from darkness to very low light. This is about compensation, but they then rise progressively. But they've already achieved a high level at the lowest light intensity. And I'll come back to this. Now, if we went back into all of the old data, which many of us had collected back in the 80s and 90s, you can see this happening there as well. So what's going on? Well, why keep fructose bisphosphatase and SBPAs relatively low activity in low light? We think the answer is they catalyze futile cycles. And if they were active in low light, you would just get a cycle for the FBPAs between PFK and FBPAs. This would burn off ATP, and this would severely impact photosynthesis efficiency in low light. So they keep the Calvin Benson cycle, these two enzymes, partly inactive to save energy wastage, prevent energy wastage. The second feature, why are the metabolites already hitting a relatively high level at the lowest light intensity? This implies a strong restriction on end product synthesis in very low light. So let's go to some fundamental property of the Calvin Benson cycle. It's autocatalytic. Its operation requires that enough triose phosphate remains in the cycle to generate ribulose bisphosphate. You can only take out what is in excess. How does this happen? There's a threshold regulation of the sucrose synthesis, also other end product synthesis. This network involves fructose 2,6-bisphosphate, a few other things, and it plays on highly cooperative enzyme kinetics so that no sucrose synthesis occurs until you hit a threshold level of triose phosphate. Then it rises sharply. And so we think this is a very general feature across all species. So summarizing up to now, our current and probably totally wrong working idea, there is variation in CDC operation. That's something we're sure about. We think there may be conserved responses, and what we've picked up here particularly is to avoid inefficiency in low light or low CO2. I haven't emphasized that. We think there are different strategies to drive a rapid flux in the Calvin Benson cycle. Now, what's the next level, which we haven't started, but I think it's for the future? This information about metabolite profiles allows us to shortlist enzymes that are likely to be better or worse in one species than another. And focusing on these suspicious enzymes, we can then look in at enzyme activity and protein abundance, transcript abundance and promoter sequences, enzyme kinetics, and of course the amino acid sequence, and try to get down to the molecular basis of this diversity. Now's the why question. Why is there interspecies variation? I was very surprised when we first saw these things going off in all different directions in the PCA. But the more I thought about it, the more I realized, well, this is obvious. And I'm sure for many people, was already obvious. So the Calvin Benson cycle is 20 billion years old. It's very old, but it's been subject to continuous selective pressure among many, many things. And here's just a couple named that John mentioned them. In the last 30 million years, this pressure, especially falling CO2, has been enough to drive the evolution of C4 photosynthesis in at least 60 different plant lineages. So there was massive selection pressure on plants. But now I'm playing on a very nice paper from David Hackman and colleagues, where they call up this climbing Mount Fiji. That's how the plants get up to the top. But what happens to the lineage if we didn't scale Mount Fiji? They had to sit down here at the bottom. Did they just sit at the bottom and sulk? Or did they actually also try to adapt in whatever way they could? And what pressure is exerted by local differences in light regime, temperature, water availability, nutrient availability, and so on. And of course, there would have been selection. Plants would have been struggling to survive. And the key point is any evolutionary adaptations of the Calvin Benson cycle would have occurred independently in each extant plant lineage. depending on its own prehistory and on all of these local conditions, so that we can expect to see large variation in how the Calvin Benson cycle operates across different species, even within species. I'd like to mention the work from Colin Osborne here, which I think is really fascinating. So I think I have a little bit more time. So I now want to tell two more unpublished stories related to Calvin Benson cycle operation in combination with carbon concentration mechanisms. One of these has to do with the Calvin Benson cycle in algae with pyrenoids, and the other has to do with the Calvin Benson cycle on the evolution trajectory from C3 through C3-4 and C4-like to photosynthesis. And I'll take this one first. So a very brief primer here. This is based on the species Flavaria, but it's similar in the genus Flavaria, but it's similar in other genuses. As you move from C3 to C4, it's thought to have occurred via various steps. After the establishment of the anatomy with the Kranz anatomy, then you developed a system where they put a glycine decarboxylase into the mitochondria of the bundle sheath. This led to the CO2 being released in the middle of the leaf and having to diffuse back outside, and it was recaptured by rubisco more efficiently. And then a little bit further on in the progression, you get a non-optimized C4 cycle with imperfect cell specificity, regulation, and properties of enzymes. And these are still extant in some, these almost living fossils maybe, are still extant in some genuses like Flavaria. So what we did here is we took the data set which we already had, and we added in C3, C4 intermediates and C4-like Flavaria species. So here we added in 2 C3 Flavaria species, and I'll actually put a dot, 2 C3 Flavaria species, 2 C3, C4 intermediates, which are the first step, a set of 3 C4-like, and then we have the true C4s out here. So what do we see? Well, first, the C3 Flavaria species lie in the middle of the other C3 species. So this was nice to see. It's another corroboration that C3 are different to C4. What was really fascinating is the C3, C4 intermediate and the C4-like species lie on the trajectory between the Flavaria C3 species and the Flavaria C4 species. The implication is that the Calvin Benson cycle adjusted progressively during the stepwise evolution of C4 photosynthesis. And this evolution was already starting, this adjustment was already starting in the C3, C4 intermediates and continued through the C4-like to the C4s. What's driving it? I'm not going to go into a lot of details here, but when we look at the weighting on the PCA, we can see that what is driving the evolution from C3 to C4 is the C3s, well, have more ribulose bisphosphate. What's happening over here? All these metabolites here are dropping. So we're looking at less ribulose bisphosphate, less FBP, less SBP, and so on, and more PGA. And this is sort of the pattern. One thing I forgot to measure, there's an outlier here. This is a Flavaria brownii. This is in the second clade of the Flavaria species. And it's thought that this evolved separately, and nothing in this clade actually evolved into a C4 species. So this may be going down a blind alley, or it may just be a different route which hasn't got there yet. Now I want to come on to the last example. This is algae with a CCM. So they basically, I've left out all the details here, but they're basically pumping bicarbonate in, gets into the pyrenoid, and here carbonic anhydrase turns into CO2 and generates a high concentration around CO2. So what we did here is Chaim Perivas had collected data for 3, in a completely different project, had collected data for 3 algae in ambient CO2 and a range of different light intensities. And so we combined this with the data from our earlier studies. We don't have the Flavarian here. What do we see? Here's the C4 species. Here's the C3 species. And all of the algae are sitting over here. Now, this is sort of interesting because they're not sitting with the maize species. They're not sitting with the C4 species. Both have a carbon concentration mechanism, but they're almost diametrically opposed in PC1. What's driving PC1? It's ribulose bisphosphate. Also a couple of other metabolites. Now we go into the original data, and you can see immediately out of this, we didn't actually need to do a PCA. We can see the The maize species, the C4 species, have very low ribulose bisphosphate. This is because they have a very efficient rubisco, they have a low rubisco abundance, and they don't have as much ribulose bisphosphate. The C3 species have an intermediate level, and the algae, all three algae, have very high levels of ribulose bisphosphate. Now, what do we think is going on here? This is a hypothesis, but I think it's a reasonable one. This is based on work which we published recently together with Fred Sarma and Luke McKinder and Annika Kuchen. The first thing in this paper that we saw is all of the Calvin Benson cycle is located outside the pyrenoid, except rubisco. That means the ribulose bisphosphate has to go in. And we suspect that these high levels of ribulose bisphosphate may be required to actually drive the rapid movement of ribulose bisphosphate into the pyrenoid. And there's a little question here. The PGA has to move out. So why don't we have high free PGA as well? In this paper, Annika, based on a lot of modeling, postulated that there may be some sort of facilitated diffusion such that the PGA is really removed very efficiently out of the pyrenoid, possibly due to charge changes. There could be other explanations as well, but we're very happy to actually see the original paper was based on Clammy, that we've actually managed to confirm this general pattern of metabolites in two completely separated, phylogenetically separated algae species.
4 minutes to go, Mark.
Yep, I'm finished. So concluding, what we hope we've done is to show that you can use profiling of Calvin Benson cycle metabolites to identify first, to see differences between different modes of photosynthesis, between C4 photosynthesis, intermediate photosynthesis, C3, and the CCMs. It's actually going to be fascinating to look what it looks like in cyanobacteria. So you can see different photosynthetic types. What we think we can also do with it is to pick up differences in how different species in different phylogenies with the same mode of photosynthesis, but are actually running or poising their Calvin Benson cycle in a different way. This is presumably reflecting the way that they've gone about dealing with the evolutionary pressure or that they've had over the entire history of the Earth, but I think particularly in the last 20 to 30 million years, and possibly even more recently, depending upon the kind of conditions that they're experiencing at this time. So I think there's a lot of variation out there, a lot of adaptation to look at, and I hope people will have fun doing this in the future. And I'd just like here to thank the members of the group who are involved, in John Lunt, Steffi Harivo, who really drove it, Regina File and Manuela Gunter, who run the machines, Jan Luca, who was involved in the rice work, Haim, the intermediate work and the rice work, Haim for the unpublished data on algae, and Marta Ludwig, who's been very important in the work on the intermediates. And with that, I'll release the screen.
Thank you very much, Mark. If you have any questions, please type them into chat and we'll call you. Eve. Eniola should unmute you so you can ask a question.
Yes, can you hear me?
Yep.
Okay. Hi, Mark. Nice talk. Thank you very much. I have a question about the comparison between rice and Arabidopsis. You showed the response to the light intensity, and you showed that rice plateaued much earlier than Arabidopsis for CBC intermediates. So I was wondering, has this something to do with I guess the ability of Arabidopsis to better acclimate to low light intensity, and could you imagine some strategy there to unravel the mechanisms Arabidopsis is using?
Yes, it might have, Eve, but it's something I didn't go into in the talk. There is a lot of very interesting work from the 1990s, and around that time, I think it's still running, that the regulation of rubisco differs, the extent to which you actually, when do you activate rubisco via the activase and also other mechanisms. And I suspect that may also lie behind it, a different way of regulating rubisco. Another very interesting point in rice, which I left out of the talk totally, but people go and look at it, is rice leaves have a different anatomy and ultrastructure. They have highly lobed leaves, and they also tend to have their mitochondria behind the chloroplasts, which form a very dense mesh on the surface of the mesophyll cells. This is work from Rowan Sage and collaborators. So what may be behind this, it may be partly also reflecting an ultrastructure, which is an adaptation, which is allowing rice to be a better recapture of its CO2. So there could be more than one thing between the rice. And this also allows rice to have high protein contents in its leaves, which is a thing I left out of the talk, but we've noticed that the protein content of these seed-free leaves does vary, which has very interesting connotations for how they're regulating CO2 movement through the stomata with Calvin Benson cycle and CO2 recapture.
Thank you. And a question from Rob Hancock.
So you've found these quite nice different adaptations and different modes of function, I guess, of the Calvin Benson cycle. So have you yet started to unravel whether any specific functional modes are associated with specific niches?
No, I mean, this is obviously a next step to go. This is correct. This would involve thinking very carefully about species, probably even closely related species, phylogenetically related species growing in different places. My time is running out, but this would be fascinating to do and get in contact. I mean, this would be fascinating to do. These experiments don't take a lot of time. So it would be possible to compare. That would be fascinating. It might also give more clues because they're very closely phylogenetically related. This might be better material to try to bore down on what are the molecular backgrounds. If I'm comparing two phylogenetic phase separated things, of course, there's all sorts of changes. But if I've got two things which are close together, this will be a much better system to go in, bore down to what are the actual changes in the promoter or the protein sequence.
Thank you.
Luke McKinder has a question.
Hi, Mark. Thank you for the talk. Really interesting about the metabolite levels in algae. Have you thought about looking at like a pyrenoidless algae, or even say Chlamydomonas with which fails to correctly form a pyrenoid? Yeah.
I mean, there's so many. I think that there are so many things that could be done here. It would be interesting to look. We have a little bit of data, but not enough to. We must dig around. I mean, to what extent do these gradients actually disappear if you don't have the pyrenoid? Because this would be a very good test of my hypothesis.
Yeah. Also, with the RU like PGA levels, so the potential that you have these pyrenoid tubules which are funneling in these metabolites, would you expect to extract these out of if they're protected by a membrane or traveling through a membrane?
Yes, we would have those the way we're doing it. We would have it. But the idea that Annika proposed is that if we have some sort of charge properties on these channels, then they would tend to drive the movement of I mean, these compounds are all phosphorylated, they're all binding magnesium, but they will have charge properties. So you could imagine the charge property which would extrude PGA very effectively is likely not to be particularly favorable for ribulose bisphosphate going in. A very simple-minded idea, but you probably can't do both. There is another possibility with respect to this, and that is that the balance between the Calvin Benson cycle and the light reactions might be shifted. So the light reactions really drive PGA reduction very, very effectively and pull the PGA down. And this would also help the movement out of the pyrenoid.
Yeah. Great. Thanks.
Does anyone else have questions? Please type into the chat if you do. Yes, Jean Panacci. Hello. Are you there?
I think he dropped off the meeting as soon as we unmuted him.
Oh, okay.
We'll try and get him back. Can you hear us, João?
Meanwhile, we've got a question from Bob Furbank.
Mark, I don't know whether it's popular anymore, but in the old days, Tony Ashton published a paper saying a lot of metabolites were bound to Rubisco, and other people were saying, well, metabolites are channeled and enzymes stick together, and the like. Would that affect the stuff that you're doing in this work?
Sure. I mean, very, very likely. That is likely to be one of the things which is underlining differences between C3 and C4 could well be just the 3 or 4-fold decrease in Rubisco abundance will not only impact on how much ribulose bisphosphate you need, but may also mean that less FBP, less SBP are binding in on the sites. Yes. I mean, another thing which I haven't touched on here at all is CP12. I think CP12, which also seems to vary in the way that it's operating between different species, and could be important, could be particularly important in low light. I think there's just a whole, I think there's a whole can of worms, or there's actually a box of chocolates, the way I would see it, which is here to be looked at. And the only thing that I think we've done is just a highlight, it's a way to highlight in a given 2 or 3 species, what's different between them. So which are the enzymes that you should go in and look at? Just in the way that back in the 80s, we used to do this to decide which of the 15 enzymes in the pathway do you go in and do all the in vitro enzymology to try to find a regulator.
Next question from Joan Pananchi.
I think he's going to type it, Nick, because his microphone's not working. So we'll move on to Howard's question just for now, I think.
Okay, Howard.
Yeah. Hi, Mark. Thank you. Just wondering, when you've done your metabolite pools for pyrenoids, do you only find PGA in them, or is there any reduction going on?
Oh, no, we haven't been able to measure the metabolites within pyrenoids. These are overall, the analysis which we published in the eLife paper, this was based on modeling by Annika. So we just tried to model with, as always in modeling metabolic pathways with assumptions. The major thing that we see is that we do have this very large pool of ribulose bisphosphate. So if you have a big pool, you have a lot of potential to put up a concentration gradient to drive diffusion. If I have a small total pool, I don't have the same potential to generate a concentration gradient to drive diffusion. For me, this is for me the major observation. The modeling was also very nice and does certainly confirm the ideas. But that's our basic observation. And I'm very happy to see that a completely different set of measurements in 3 algae independent came to the same result. And I could basically identify an alga with, I would say I could do classification. It would be a game in terms of bioinformatics to say, can we classify species without knowing what kind of photosynthesis they're doing based on the metabolite, Calvin Benson cycle metabolite profile, in exactly the same way you can do this with delta 13C.
Well,
but much more deeply, of course.
Just a plug for Miriam's paper. We've got a nice one showing the distribution of the CCM across the green algal phylogeny. So you've got plenty to choose from.
Yeah.
Okay. In the absence of any other questions, I've got one. Mark, have you looked at any bryophytes, mosses, liverworts, and the kind of liverworts that have pyrenoids? You're muted.
Okay. Okay, now I've got myself. Can you hear me now?
Yes.
Yes. No, we haven't. Time is the essence. It would be very, very interesting. Maybe John is listening into this. I think we are starting to do a bit of stuff on Mercantia. It would be very interesting. Also, obviously, it would be interesting to go into gymnosperms and so on. There's just too much there to do. Yeah, very good idea.
Sure. Okay, I'm going to read out this question now. Considering that mainly in protocrants, some anatomical changes are already present in preparation to C4 pathway, as increase in bundle sheath cells area compared to mesophyll cells, and considering the bundle sheath are still not photosynthetically active, could this influence the metabolic analysis you have performed?
I think the answer there is your guess is as good as mine. There may well be differences. I think my answer there would be, until we know more about how much difference there is really on a microphylogenetic level, it would be a little bit dangerous to interpret a change as due to the protocrants on its own. But that's not a very good answer. Yeah, so I think the answer is yes, there might be, but how would you be sure it was due to the protocrants? There is an approach which we could potentially take in the next years as developmental biologists are moving to have the genes, the transformation factors which are controlling the bundle sheath size. If you could actually engineer a plant with larger, more metabolically active bundle sheaths, then that might be a more direct way to address this question, or at least to back up any study of existing wild species.
Excellent, thanks. I think we'll have to draw the questioning to close here. Thank you very much, Mark, and fantastic to see what you can do with a triple quad mass spectrometer.